Dear grade 12, this is going to be the second of 3 video lectures on meiosis. Um, let's get into it. So, in the previous video, I introduced you to some terms: diploid, haploid. Introduced you to meiosis and mitosis, understanding the differences. Here we're gonna go a little bit more in depth. We're gonna have a quick recap on mitosis. We're gonna have an in-depth look at meiosis, what is the important things we need to know about meiosis, and yes, all of those fun stuff. But firstly, we need to obviously get started with what is cell division, because in mitosis and meiosis we know that there is some sort of cell division happening. Now, cell division is the time during which the cell grows, replicates genetic material, and divides and this is obviously known as the cell cycle. But cell division is it's the period in which the cell grows, it's the period in which the cell replicates genetic material, and it's also part of the cell cycle. Now, in the cell cycle, we have two phases. We have the interphase and we have the dividing phase. Now, remember the interphase we discussed in DNA as well. Now, the interphase is the period between two successive cell divisions. What does that mean? Successive means after each other. So there would be a cell division, you'd have the interphase, and there you'd have the cell division. Right? And in the interphase, we know DNA replication takes place within the interphase. Right? And that's one of the important things that we need to understand when it comes to the interphase. But if we're looking at meiosis or mitosis, and we need to identify the interphase, we know that it's the interphase when the chromatin network is visible. Now remember, we obviously know that the chromatin network is that ball of cells that we find within the nucleus. And we can see that over here. Because the chromatin network is visible, we know that it is the interphase, because only in the interphase, only in that phase where DNA replication is taking place and other metabolic processes are taking place, then we know, obviously, then we know it is the interphase, right? And also we will see that there will be one pair of centrioles in the centrum here. So it will look like a normal cell, but what gives it away is the fact that it's the interphase is always the chromatin network. Always keep in mind that it is the chromatin network. Now, there are important things and events that we just mentioned, one of them, that takes place in the interphase. And we didn't know it DNA replication takes place. DNA replication is the process by where we go from a single strand to a double strand. And this double strand is always joined by what is known as a centromere. Right? That is DNA replication, and DNA replication aids in doubling the genetic material so that it can be shared by new cells arising from cell division. And this is what DNA replication does. Right? Meiosis as well. This is a similar understanding to meiosis, but DNA replication is there to ensure that the mother cell and the identical daughter cells, IDC, identical daughter cells, share the same genetic material. Right? That is the event, that is the importance of DNA replication taking place, to ensure that all our cells share the same genetic material. Now, if we're going to quickly look at mitosis. Now, mitosis, we're just going to have some revision now. Mitosis is the division of somatic cells. In the exam, they can ask you what is somatic cells. All you say somatic cells is body cells. Right? You can just write body cells and you will get the mark. You don't have to still write somatic cells or body cells. You just say body cells. If they ask you what are somatic cells, your answer would just be body cells. And the purpose here of meiosis is to produce new cells for growth, repair and damage of tissues, and asexual reproduction. Last thing at 11, when we did bacteria, and when we did fungi, and when we did protists, and all of those in the microbiology, right? All those microorganisms, we normally refer to something known as binary fission. And that was asexual reproduction, right? And that is what mitosis also assists with, right? Asexual reproduction. Also, when you have a scab and as the scab is healing, it forms if you have an injury and the scab forms, that is mitosis taking place, because under the scab what is happening is the on your cells being replicated in order to make sure that skin reappears. That is mitosis, the repair of damaged tissues. And skin, and the skin being an organ, is obviously comprised of tissues. But if we just quickly look at the processes of mitosis, so here we would know that prior to mitosis, each chromosome makes an exact duplicate of itself. The chromosome then thickens and coils. That we know is the chromatin network, which is thickening and coils, right? And then when we move on in the prophase, right? Remember, the chromatin network first looked like that. But because of the thickening and the coiling and the replication that took place, right? It now appears as chromosomes, right? As single chromosomes. Now, in the early prophase, the centrioles, which have divided, form asters and move apart, right? The centrioles divide and they're going to go to opposite poles, and they're going to form the spindle fibers in between, right? And in meiosis, it's a similar thing that takes place, the spindle fiber formation. Spindle fiber formation is there to ensure that the chromosomes are pulled to opposite poles, to ensure that when cytokinesis takes place, that each new cell that is formed has a chromosome. And the nuclear membrane starts to disintegrate, the nucleolus starts to disintegrate, the nucleoplasm changes to a jelly-like liquid, so that because if the nuclear membrane, remember this is the nuclear membrane around you. Now, if the nuclear membrane disintegrates and the nucleoplasm, the fluid membrane in which these chromosomes are situated, if it doesn't change, so like these things are going to float out of the cell. So the nucleoplasm changes to a jelly liquid to make sure that these chromosomes stay in place. Right, to make sure that the chromosomes stay in place. Now, in the late prophase, the centrioles and asters, they move to opposite poles, which is just what I said. They move to opposite poles. The nucleus and the nuclear membrane have completely disappeared. And what forms between these, they say asters, but we can just say centrioles. What forms between the centrioles is called spindles or spindle fibers, right? And what attaches the chromatids of the single chromosomes to one another, right, of the chromosomes to one another, is called the centromere, right? So please understand the difference between centriole, centromere, and chromatid and chromosome. Now, the double chromosomes, right, their centromeres attach to spindle fibers, and they line up in the midsection in the metaphase, right? Remember, mitosis is PMAT, right? Prophase, metaphase, anaphase, telophase. Now, in the metaphase, these chromosomes, these double chromosomes, I said single, but I just make sure that I'm supposed to be saying double. The double chromosomes, they go and lie on what is called the equator. So I don't have enough space to write, but they go and lie. Okay, they go and lie on the space known as the equator, right? The imaginary line that divides the cell in half, right? And this lying on the equator is preparation for it to be pulled apart, but it's also preparation for cytokinesis to take place, for that invagination to occur over here, right? For the cell to completely divide and form two identical daughter cells, right? But the centromere, the centromere of the double chromosome attaches to the spindle fiber there, so that it can be pulled to opposite poles, right? These are poles. It's a pole here and a pole here, north and south pole, to pull to opposite poles. Now, here in the early anaphase, remember we said it's PMAT. So in the early anaphase, the centromeres half, or they split. Half the chromosome moves to one pole and the other half moves to the other pole. Remember, so they first looked like this. Right? Now they're going to move to opposite poles. So they're going to look like that now, as they are being split. And as they are moving to opposite poles. Right? This is going to come to that pole and that one's going to come to that pole. Right? Because the center, remember the centromere is split. So half, half. Right? In the late anaphase, the chromosomes have almost reached respective poles. The cell membrane begins to pinch at the center. Right? So, P mat again. In the anaphase over here, right? In the anaphase here, we see the beginning of cytokinesis. The halving of the invagination, or the halving effect that is going to take place with the cytoplasm. Right? The cell membrane begins to pinch at the center. It starts to invaginate. Cytokinesis starts to take place. Right? The cell membrane completes constriction in the telophase. The nuclear membrane form around the separate chromosomes. So now these chromosomes, the nuclear membrane is going to reappear. The nucleolus is going to reappear. We still have this invagination, the cytokinesis effect. Cytokinesis is still taking place over here to make sure that this is going to be split in half. And this is part of the telophase. This can be known as the early telophase because it is not complete yet. Right? At the completion of mitosis, so this is at the end of the telophase. At the end of the telophase, there are two cells with the same structure, the same number of chromosomes as the parent cell, and that is what we said in the beginning. The significance of mitosis is to make sure that the mother cell is identical to the two daughter cells. So we started off with the mother cell with 46. We're going to end up with 46. If we start 48, because of mitosis, we must end up with 48. Right? And that is the repairing and the growth that is taking place. So that was just your revision of mitosis. Now if we're going to look at meiosis, the main difference between mitosis and mitosis deals with body cells and meiosis deals with sex cells or gametes. The formation of gametes, where we're going from diploid to N to N. This is the halving effect, reduction division. Meiosis is also known as reduction division. It can also be called reduction division because there is a halving effect happening. We're going from 46 to 23. We're going from 4 to 2. We're going from 8 to 4. We're going from 6 to 3. We're going from 16 to 8. There's a halving effect that's taking place where the daughter cells at the end of meiosis 2 will only have one set of chromosomes. Right? And therefore they will not be identical to the mother cell. Remember, in meiosis, the mother cell was identical to the two daughter cells. But in meiosis, that is not the case. The mother cell might have had 46, but here we will have 23 and 23. Right? That is the reduction division that is taking place. That is meiosis that is taking place. And to understand this, we need to look at the life cycle of a human. Right, the life cycle of a human. If you look at the life cycle of any organism for that matter, and you do this. But let's look at the life cycle of a human. If we look at the life cycle of a human, the paternal parent or the male will have 46 chromosomes. The female will have 46 chromosomes. Right, XY and XX. The male gametes or cells will undergo meiosis and the female will undergo meiosis, and they will produce 23. And the significance of this halving effect, remember this is the sperm and that is the ovum. The significance of this halving effect is so that when the sperm and the ovum fuse during a process known as fertilization, that the zygote that is formed will also have 46. Because if meiosis did not take place, and I'll do this in red, if meiosis did not take place, it means that the sperm would have 46 and the ovum would have 46, which would mean that the zygote would have 92. And this is a completely different organism. It's a completely different organism, right? and then mitosis will take place on the zygote, right? And you will eventually start growing, and each cell will have 46. And then eventually that same thing, whether the zygote is male and the zygote is female, and that zygote or that human goes to sexually reproduce, the same thing is going to happen. And this is the continuous cycle, the Lion King circle of life, to be, if you think of it that way, right? It's a continuous cycle that is consistently taking place. But meiosis is divided into two distinct phases. It is your first meiotic division and your second meiotic division, but there are differences. In your first meiotic division, there is the halving of the chromosome number from diploid to haploid. The genetic material is exchanged to produce genetic variation, and this is through a process in prophase 1, which is known as crossing over. Right, and we're going to look at that at a point known as the diastema. Right? That is meiosis 1. In meiosis 2, it's like a normal mitosis where the chromatids separate from each other. So meiosis 2 is exactly this. It's like mitosis. All you're doing is you're separating chromosomes, it's separating chromatids, and making sure that there is going to be 4. Remember, you're going from the definition of mitosis, or some textbooks use this definition. The definition is rather, not a fan of the definition, but it is when division takes place and we have 4 different haploid. So you went from a cell that was 2N, and the end product must be 4, depending on how much you started with, 4 different haploid. Right, so you went from diploid to haploid. Right, and meiosis 2 is just to ensure that the 4 are haploid. They already become haploid here. Here it's just to ensure that the copies are being made. Right, so that we can have 4 haploid. Note, both happen as to PMAT. Before meiosis, replication takes place in the interphase. Just note those two important terms or important notes. So now we're going to look at prophase 1 first. Let's start with prophase 1. So I'm first going to tell you, how do we identify its prophase 1? Right, we can see that spindle fibers have formed, but they haven't moved to opposite poles yet. We can also see that there is a homologous chromosome. What does homologous mean? One comes from the mother, one comes from the father, paternal chromosome, maternal chromosome, and they are touching each other at the point known as the diastema or chiasma, whichever way you want to say it, so you can remember it. But at the diastema, which is the point of contact where genetic material is being, or genes are being exchanged by a process known as crossing over. But let's quickly look here. Let's look at the words. So they say chromosomes shorten and become visible as 2 chromatids joined by the centromere. Homologous pairs of chromosomes are now visible. Homologous, one from mother, one from father. Nuclear membrane, nucleolus disappears, similar to mitosis. Spindle fibers are to form. Chromatids of each homologous pair touch at the point of contact, or the point at which they touch is known as the diastema. DNA is crossed over at the diastema, and this crossing over of DNA here the process is known as crossing over. This DNA that is crossed over, the process is known as crossing over. And we're going to look at crossing over a little later. But basically what is happening is, if you have your maternal chromosome here, and you have your paternal chromosome over here, remember they are going to touch. So at this point here where they are touching, right, the point of contact, that is known as the diastema. This is a homologous chromosome pair because you have maternal and paternal, right? And what is happening at the diastema is the exchange of genes. So set of genes is going to the mother from the father, and a set of genes is going to the father from the mother. And this is to ensure something called genetic variation. This is to ensure that offspring are genetically different. This is to ensure the survival of offspring. This is to ensure that natural selection takes place. There are a lot of definitions, and not a lot of definitions, but there are a lot of explanations for as to why crossing over must take place. But we're going to look at it a bit later. Spindle fiber continues to form. Right? Now, here in meiosis, I think I did correct. Yes, I did. I'm just checking. So now here, that was prophase 1. Now we're going to look at metaphase 1. So now we can see that what is the result of crossing over. Can you see this would be the result of crossing over. That is the result. 1, this is showing us that crossing over has taken place. This is showing us that there was a swapping of genes. That's showing us there was a swapping of genes, right? How do we also know this metaphase? They are lined on the equator. The centromere is attached to the spindle fibers, right? The dotted line tells us that this is the equator, right? That is how we identify that it's metaphase 1. Metaphase 1, the spindle extends across the whole cell. Homologous chromosomes, now it's not a pair anymore, right? It's not a homologous pair. It's now homologous chromosomes. It's fine enough. So homologous chromosomes line up along the equator of the spindle in their homologous pairs, right? So it's homologous chromosomes lining up as homologous pairs. Understand that. One chromosome of each pair lies on either side of the equator. The centromere of the chromosome attaches to the spindle fiber. And we know from our knowledge of meiosis that this attaching to the spindle fiber is in preparation for it to be separated and pulled to opposite poles. Right? That is the preparation that's taking place. Anaphase 2, we can now see that this pulling of the spindle fibers, this contraction of the spindle fibers to opposite poles, right? We can see that is taking place where they are now pulling homologous chromosomes that are in their homologous pairs. They are pulling them apart, and now they will now be known as full chromosomes. The spindle fibers shorten and pull full chromosomes to opposite poles. Now they're not pulling homologous pairs. You need to understand why I say that. Because if you are going to say in the exam that they are pulling homologous pairs, you are saying this. Let me illustrate what you are saying. You are saying this. That just pretending that it's a nice drawing. You are saying that that 2 sets are being pulled together. That is not the case. It is full chromosomes. You need to understand the difference in the wording. That is very important because it changes at NSC level. Right? Full chromosomes. The spindle fiber shortened. That's why I took this part out here. That part I took out here. The spindle fiber shorten and pull full chromosomes to opposite poles of the cell. Now, what I want to tell you here with my meiosis, the only way that you're going to understand meiosis is by doing past paper questions. You need to be actively reinforcing the knowledge, right? You can't just go in and memorize the table. The table's not going to help you. You need to be able to understand what is happening by doing past paper questions. When we come to the telophase, they call this constriction. It's not constriction, it is called cytokinesis. Right? It's called cytokinesis. Don't make that mistake and write constriction because this comes from mind the gap. It is cytokinesis. The full chromosomes reach the poles of the cell, right? Remember, they now have reached their various poles. This has gone that way, that has gone that way. They've now reached their poles, and when they reach their poles, each pole has half the number of chromosomes present in the original cell. That is true because here we had 4 chromosomes present. Remember, there was 4. Now here at the end, each cell has 2. This cell had 4 here, and now each cell has 2. And this is that effect of going from diploid to haploid. That is reduction division. There we can see the reduction division taking place. In our cell it would be different. In our cell we would have had 46 there and we would have gone to 23. That is the reduction division that is taking place. The cell membrane undergoes cytokinesis and divides the cytoplasm in half to form two cells. But remember, here we had one cell, now we have two cells, but the significance is that one cell had four, but these two cells have the significance is that these two cells, remember that this is now a cell and that is a cell, but the significance is that these two separate cells contain two chromosomes. That is what we're looking at. We're looking at the halving of the chromosomal number, not the doubling of the cell number. That is not significant to us. We're looking at the halving of the chromosome number, that we went from 4 chromosomes to 2 chromosomes. That is the significance of reduction division. Also, I just want to go back to metaphase 1. Right, this lying on the equator is also very important for genetic variation. It is called random arrangement. In metaphase 1 and metaphase 2, the process of random arrangement, because that one could have also gone and lined here, this one could have gone and lined there, they could have swapped, but instead they lie like this. This random arrangement is to ensure even more genetic variation, is to ensure even more strong environmental characteristics of the offspring, to ensure that the offspring have a greater chance, the greatest chance of survival. Crossing over, random arrangement, we're also going to look at something called random fertilization. Those three things are there to make sure that genetic variation is so strong that you will look the same like your brother and sister, but yet you will be so different. If you are twins, or in general, you can look the same, but you will be so different. You will have different characteristics, and that is why it is so significant to make sure that the offspring has the greatest chance of survival, to withstand environmental resistance, to make sure that natural selection takes place, to equip the offspring with everything that is necessary. Right. Let's look at the second meiotic division. Now, in the second meiotic division, each cell formed during meiosis 1 now divides again. A spindle fiber forms in each of the new cells. Single chromosomes are visible. So remember, we had 2 cells, so now this is the 2 cells over here. Now we can see that a spindle is developing in each of them. Right, in this cell, in cell A and cell B, we can see that spindle fibers are developing. We can see if we bring down like that, we can see that this grouped together here. Right? And these are called single chromosomes. So don't make the difference. Right? Let's just quickly look here. Here they are called homologous chromosome pairs. Here they are called... Here we call them homologous chromosomes that occur in homologous pairs, or you can, in fact, you can actually just say homologous pairs is fine. Here we call them full chromosomes. Here we also call them full chromosomes. Here the name changes to single chromosomes that are now visible. Now here in metaphase 2, please remember the important process that is happening in both cells here of random arrangement. Right? And also please note, in prophase 2, there is no crossing over taking place. Crossing over only happens in prophase 1. There is no crossing over taking place. Let's read. Individual or single chromosomes line up at the equator of the cell with the centromeres attached to the spindle fibers. Please, you need to include in metaphase 1 and metaphase 2 the significant you need to include this sentence here because of the centromere attaching to the spindle fiber, because it is significant in the pulling apart effect that's going to take place. Right, now in anaphase 2, here we can obviously see that they are being pulled apart, and here we can already see the formation of the 4 haploid that is going to take place. That's going to be A, B, C, D. We can already see that is going to take place. Right, the spindle fibers start to contract, the centromere split, and chromatids, not daughter, we don't say daughter chromosomes anymore, that is, the centromere split and chromatids are pulled to opposite poles of each cell. Right, so here can you see here, that was our centromere, but now our centromere splits So that this chromatid can be pulled that way and the other chromatid can be pulled that way. And here we can already see the formation of our, as I said before, of our 4 haploid, right? Now if we look at the telophase, the chromatids reach the opposite poles and the nucleus starts to form. The cell membrane of the cell undergoes cytokinesis, and the cytoplasm divides into 2 cells. 4 haploid chromosomes are formed, not daughter cells. 4 haploid chromosomes are formed. Each cell has half the number of chromosomes of the original cell. That is true, right? Remember we had 4, now we have 2, 2, 2, 2, right? The cells are genetically different from each other. How do we know that they are genetically different from each other? Here we can see that this whole thing is light. Here we have dark and light. Here we have dark, more light. Here we have fully dark. So that is showing us genetic variation. So depending on which sperm cell reached there first, which ovum, right, will either get one of these sets of characteristics. All of them have different characteristics, different traits, right? And that is why we say that they are genetically different. And here I made a nice note of the names of that is taking place in the different phases. In prophase 1, it's homologous chromosome pairs. In metaphase 1, it's homologous chromosome pairs. Anaphase 1, telophase 1 is full chromosomes. Prophase 2, metaphase 2, it is single chromosomes. Anaphase 2 and telophase 2, it is chromatids and chromosomes. These are the different names. Now let's look at my favorite part now. Crossing over in prophase 1, which I did explain already. Now crossing over is there to introduce genetic variation in gametes, right? And this can lead to favorable characteristics which will therefore affect the chances of survival of the organism, which is what I said. Right? If in the exams they ask you the significance of crossing over, you will say this is your answer. Crossing over introduces genetic variation in gametes, which may lead to new favorable characteristics. Therefore, it affects the chances of survival of the organism. Now, here is 2 ways that I saw in which the questions can be asked. Describe the process that caused the inheritance of a combination of both parent characteristics in the chromatids. What is this question asking? They're asking for the process that caused a chromatid to have some information from the father, but also some information from the mother. And the only thing that could have caused that is that crossing over, the swapping of genes, and this would be your answer for that. If they ask you the significance of crossing over in natural selection, this would also be your answer. That is your answer. Right? Let's quickly look at random arrangement. Random arrangement of chromosomes in metaphase 1 and 2. Now, chromosomes line up. If they ask you for the significance of it in exams, if they ask you what is happening, you will say chromosomes line up in the equator in different ways on the equator of the spindle. This allows for gametes to have different combination of paternal and maternal chromosomes. Because what are they saying here? It allows, so instead of having 2 green together like that, I can maybe have, because of random arrangement, I can have a green and half blue with a fully green. Something like that. That is what random arrangement is. It is to make sure that there are different combinations. Not everything is the same. Because if everything is the same, you're going to have, and you have 4 children, they're all going to be identical twins. They're all going to look the same. They're all going to have the same characteristics. They're all going to have the same survival traits. That's not what we want. We want organisms to bring variation. We want different people bringing different chances of survival, different organisms, and that is the importance. Random fertilization here, it is chance combination as to which sperm cell will fuse with the egg during fertilization. What is this that they are saying? It's a lot of words. And they don't normally ask random fertilization, but we must include it to show genetic variation. Random fertilization just means that you don't know who your future partner is going to be. You don't know okay, I can say partner, but you don't know who your future husband is going to be, who your future wife is going to be. That is completely up to chance, right? That sperm and that egg that will fuse one day is completely up to chance. God's willing, if you want to have children, that is completely up to chance. That is random, right? You don't know if you're gonna meet your husband or wife in a club, in the church, in your street, in your school. It doesn't matter. But the point I'm trying to get across is it's chance combination as to which sperm cell will fuse with which egg during fertilization. It is totally up to chance, right? Unless you've planned it out so diligently, like a sociopath, that you knew since the beginning who it's gonna be, then it won't be that random. But in most cases, it is quite random. And this year brings a unique gene combination. It introduces a shuffling of traits, and also it produces great genetic diversity or genetic variation. Now, let's just quickly look at similarities between mitosis and meiosis. Both processes are cell division processes. This is how you would answer it in the exam. Both require DNA replication to occur during the interphase before the division phase takes place. Both show the following phases: prophase, metaphase, telophase, and anaphase. That is the similarities. I don't think I need to explain that. The differences between let me just wait for this thing to focus. There we go. Mitosis occurs in body cells, or we can also say somatic cells. Meiosis occurs in the reproductive organs. The chromosome number remains constant. The chromosome number is half. Here, every cell is 2n here, or we can say diploid. Every cell is diploid. And here, every cell is haploid. Here we know that 2 cells are formed, right? Mother to daughter. Here we know that 4 cells are formed, right? Mother to daughter to daughters also separating, and that is the 4 haploid, genetically different. Cells formed at the end are genetically identical to one another and the parent cell. The mother cell is identical to the 2 daughter cells. The 2 daughter cells are identical to the mother cell. Here, cells formed are genetically different from each other and the parent. So this one is like that. I can actually draw it in all in different colors. Blue, black, but the original here could be red and blue, right? So they are all genetically different. They are not the same. Crossing over does not occur in mitosis. Crossing over occurs in meiosis. Only one division takes place. Here we have 2 divisions taking place. No pairing of homologous pairs of homologous chromosomes. Homologous chromosomes arrange in pairs during prophase 1. Also, difference between prophase 1 and prophase 2 is that in prophase 1, you can write this down, just my words verbatim. Prophase 1 crossing over takes place. Prophase 2 crossing over does not take place. In prophase 1, we see homologous chromosomes. In prophase 2, we see single chromosomes. In metaphase 1, right, we see homologous chromosome pairs moving to opposite poles. Right, in metaphase 2 over here, we see we see single chromosomes moving to opposite poles. In anaphase 1, we see full chromosomes moving to opposite to no, I'm talking nonsense here. Sorry. In metaphase 1, we see homologous chromosome pairs lining up at the equator. In metaphase 2, we see single chromosomes lining up at the equator. In anaphase 1, right, we see full chromosomes being pulled to opposite poles by the spindle fibers. In anaphase 2, we see chromatids being pulled to opposite poles via the spindle fibers, right? That's anaphase 2. In telophase 1, we see full chromosomes, and we see two genetically different haploid cells, right? In telophase 2, we see 4 genetically different haploid cells. We also see chromosomes instead of 4 chromosomes. So that would be the differences between the phases of meiosis on its own, basically. I don't think I included anything else here. In the next video, we will just be doing some exam-based questions, but seeing that they're giving me blank paper here, let's just quickly do a quick recap. Right, I just want us to remember that meiosis is PMAT. Why is this thing hacking? Is PMAT, and then mitosis is PMAT, but meiosis is PMAT 1 and as well as PMAT 2. Also understand the differences between PMAT 1, PMAT P1, P2, M1, M2, A2, A1, T1, and T2. Understand the differences between that. Understand the differences between mitosis and meiosis. Understand that crossing over takes place here, random arrangement takes place here. How would we describe crossing over? How would we describe random arrangement? Here it is homologous chromosome pairs, here it is homologous chromosome pairs, here it is full chromosomes, there it is full chromosomes, here it is single chromosomes, here it is single chromosomes, here it is chromatids, here it is chromosomes. Please understand the difference in the names. Right? Here it is 2 haploid, here it is different, genetically different. Here we have 4 haploid, that is genetically different. If we start with 8 here, we need in each, if we start with 8 chromosomes here, we need to have 4 chromosomes over there. So halving and reduction effect, we have to have 4 over there. Right? So please understand the difference. Here I'm referring to cells, 2 genetically different cells. Here you have 4 genetically different haploid cells. So just understand all of those differences. And then in the next video that I do, we will be discussing abnormal meiosis first. And then in that same video, we will do some abnormal meiosis questions because it's very hard to understand. And I'm going to break it down, make it simple for you to understand. And then we're also going to just quickly do some basic identification of structures and describing crossing over, describing random arrangement, and drawing, as well as also differences between phases.